Hello, everyone. My name is Tucker Hallmark, and I'm the Manufacturing Manager of Lipidomics at Avanti Polar Lipids. Thank you for joining us today for part one of our three-part webinar series, Exploring the Lipidome. I'm pleased to introduce our inaugural speaker for this series, Dr. Maria Fedorova. Maria studied biochemistry at St. Petersburg State University in Russia, and she obtained her PhD in the Faculty of Chemistry and Mineralogy at Leipzig University in Germany, where she then worked as group leader for the Institute of Bioanalytical Chemistry. In August 2021, Maria's group moved to the Center for Membrane Biochemistry and Lipid Research at TU Dresden. Maria's research is focused on the development and implementation of lipidomics and bioinformatics solutions to address complexity and plasticity of lipid metabolism in a variety of biological systems. In particular, her research aims to gain a deeper understanding of pathophysiology behind metabolic and inflammatory disorders, including obesity, insulin resistance, type 2 diabetes, and cardiovascular disease. Maria's talk today is entitled Molecular Profiling of Human Tissue Lipidomes, Challenges and Perspectives. During the presentation, please insert your questions into the chat or the Q&A box, and we will open for questions and discussions at the end of her presentation. And with that, I will turn the floor over to Maria. And thank you so much, Maria, for sharing with us today.
Thank you very much for introduction, and thank you very much for the invitation. So I'm just sharing my screen. And do you see it fine?
Yes, we do.
Okay, good. So today I'm going to talk about molecular profiling of human tissue lipidomes, and I want to share with you our experience, so the challenges we had to face by doing that, and then I'm really looking forward for some discussion to know your opinion about certain technical questions. So, but first, let me see how to switch. One second. Yep. So why actually we want to look for the human tissue lipidomes? Yeah, I guess we all know that lipids can be a very useful readout of human metabolism. And from this kind of cartoon, which you can see in very different variations in the textbook in biochemistry, we all know that lipids are transported over our body in the different types of lipoproteins, and some of them have more of triacylglycerols, some have more cholesterol esters or phospholipids inside. And that is pretty much reflecting what is measured nowadays in the clinical lipid panel, which everybody can get as a test in the physicians. So we can measure total lipids, serum total cholesterol, HDL, LDL cholesterol, total TGs, or phospholipids. So in principle, in the year 2022, in the terms of a population-wide diagnostic, we are not far away from those kind of cycles. Yeah, so we can say we have more yellow, we have more orange, or we have more blue. But lipid metabolism, of course, is much more diverse than that, and we can learn much more information by looking at the lipids in more specific ways. And modern mass spectrometry-based lipidomics is actually a technique which provides us access for the identity of lipid species and even lipid molecular species in a complex biological matrices. So we can use liquid chromatography to separate our lipids of interest based on the certain physicochemical properties. And we can use mass spectrometry and tandem mass spectrometry for identification. So we can look at the lipids and lipid molecular species as it's shown here. So we see with 36 carbon and 4 double bonds in the acyl chains, but even in the lipid molecular species level, when we can clearly differentiate which of the fatty acids this phosphatidylcholine is carrying around. Moreover, now we also mastered methods where we can quantify lipids and distinguish lipid quantities in disease versus, for instance, healthy conditions. We can do it in a relative quantification way where we can say, okay, this lipid is twofold increased in the certain conditions, but we also can do it now in the absolute quantification or semi-absolute quantification way where we can report a real concentrations. And this technological development, of course, provided us with a much higher level of information. So in the majority of the cases when we do high-throughput lipidomics profiling in certain biological samples, what we see at the end is that low or high level of certain lipid are associated with a certain conditions. So what we look at is a more like observation of correlation biomarkers when we can correlate or associate increase in certain or decrease in certain lipid species with certain pathological conditions. But usually I ask myself a question when I get such kind of data that, okay, I can see these differences, but what does it mean? And in order to understand and to come from these lipid fingerprints where we can see that something is going up and down and can be a wonderful biomarker, to more mechanistic understanding, we have to look at the lipid metabolism in a more systematic way. So we have to map these changes to the pathways of lipid metabolism and understand what is really going on in the given biological systems. So lipids represent the signatures of human metabolism, and this kind of concept, when we divided lipids into energy metabolism, compartmentalization, or signaling, these borders become more and more blurred nowadays. So we understand that lipids are actually involved in many different reactions and many different functions. And what is more importantly, what makes them very special in terms of different biomolecules, that in majority of the cases, they act as collectives, as functional units via their cooperative action. And what we really expect nowadays from the lipidomics workflow, or to find this answer, is a capability to map the alterations which we see using our lipidomics approaches to the metabolism of lipids and connect it to the certain functionality. So we can see a flux of lipids in the system and explain it through some biochemical pathways. That would be, of course, an ideal situation. In order to do that, of course, we are still not there and have to move further on. But one way which would support this kind of mechanistic understanding is the generation of reference lipidomes of human tissue. Because in reality, how much do we know about lipidomes of different tissues? Yeah? So we know pretty good now human blood plasma lipidome. Many works were done on that level, and but further than that, there is no systematic profiling of different human tissues performed so far. So that will help us address the specificity of lipidomes remodeling, not only in a tissue dependent, but also we have to look at the age, gender, ethnicity dependent manner. And finally, that will provide us understanding how we can use blood plasma lipidome, which is, of course, would be the best source of those markers, as a readouts for the tissue dysfunctions. So those tissue reference lipidomes would represent qualitative and quantitative inventory of lipid molecular species in particular organ or tissue in physiological and then pathophysiological conditions. So I guess many of you use this wonderful resource, which is called the Human Protein Atlas, where you can just type the name of certain gene and look the distribution of those transcripts or the proteins in the different human tissue in gender-specific ways. So maybe one day we can imagine that we will have something close to the human lipid atlas, where we can also see those lipids distributions in the different tissue organs and on the different levels. So those tissue reference lipidome will provide us readout for this specificity of lipid metabolism in particular organs and tissues, because obviously we know that brain is very different from liver, but how is that defined on the lipid level? So, and knowing that we can look for the lipidome plasticity and remodeling, that is also important for the clinical translation, because that will allow us to correlate these reference values and intervals, which we can see in the blood plasma, to the readouts of the different organs and tissues. And from the systems biology way, of course, that would be a great help to look at the lipids by using, for instance, lipid set. enrichment analysis or perform big data integration via genome scale metabolic models. So with that in mind, a few years ago, we started a project where we wanted to look in the lipidome changes in obesity. So there is no wonder that obesity is a risk factor for the public health, and this is one of the pandemics which we are facing nowadays. Unfortunately, obesity doesn't come alone just with increase of the body weight, but it comes with many different metabolic complications. So cardiovascular diseases, fatty liver, diabetes, and so on. And within the obese individuals, what we know for now that there are different phenotypes, and some people develop obesity in what is called metabolically healthy way, and some of the people, unfortunately, majority, go in metabolically unhealthy obesity. The difference between those two conditions include differences in insulin sensitivity status and the amount or volume of visceral fat, and also in the hypertrophy of adipose tissue, as well as the levels of inflammation. And with the general trend moving towards personalized medicine, if we ever gonna to understand and actually use our knowledge on the adipose tissue biology towards this direction, we have to understand this heterogeneity on these different metabolic phenotypes, but also between different depots of the white adipose tissue, and then gender, age specificity, diversity of metabolic complications, and individual differences in the lipid metabolism in white adipose tissue. So coming back to the analytical perspective of what we did here, so we had access to very nice set of biopsies provided by Matthias Bluer back at Leipzig University, where they have very nice biobank built it up for the obesity research. So we had a paired biopsies from subcutaneous and visceral fat from the same individuals, and we also had some samples from lean individuals. And the obese cohort was subdivided into two phenotypes. So some of obese individuals were insulin sensitive, so healthy obesity, and some of them were insulin resistant. So when we started to look into the planning our analytical approaches into looking into the adipose tissue lipidome, of course, this tissue is very complex for lipidomic analysis, and mostly due to the fact that, logically, the majority of lipids present in white adipose tissue are triacylglycerols. And this is, of course, a function of this tissue to store them, but for the analytical science, it's a little bit challenging. Yeah? So we have more than 98% of lipids in white adipose tissue represented by TGs. And if we want to get access to the polar lipids, which of course also very important for the biology of the tissue organization, so such as PC, PE, and so on, those lipids are only 0.3% of the total lipidome. So that was an analytical challenge which we addressed on the different levels. So we went through optimization of this tissue-tailored lipid extraction, fractionation, and analysis, because we wanted to get really this non-discriminative coverage of all, preferentially all, lipid classes and subclasses. So after testing different type of extraction methods, we came up that we proceed with a Folch-based method for extraction. But if we do that, we of course very efficiently extract our triacylglycerols. And if you could see on the right-hand side the picture from the TLC, yeah. So if we do that, we don't see actually anything. Okay, now it's pointer. I'm not sure if you see the pointer. So we will see only those triacylglycerols on the TLC. And we went afterwards for the special enrichment step where we made an second round of liquid extraction to partition our polar and unpolar lipids. Yeah, and you can appreciate again on the right side on the TLC that after the second round of liquid extraction, indeed we started to get access to this 0.3% of our lipids, polar lipids in the white adipose tissue. So then we went through quite intensive data acquisition and data analysis workflow, which are summarized on this graph. So we use different types of chromatography, different types of mass spectrometry to acquire a lot of LC-MS data from pooled samples at this stage. And we afterwards went for the data interpretation. So just a few words about that. So in this case, we worked with the pool samples where we pooled our lean so the adipose tissue from lean individuals or obese individuals based on the phenotype or based on the depot, so subcutaneous and visceral. And for these different extracts which we had here, for instance, for the total lipid extracts, which was basically all triacylglycerols, we performed different type of mass spectrometry-based analysis using reverse phase chromatography, mostly on the C30 columns with a positive negative data-dependent acquisition. We also used this Acquire X workflow, which allows us to dig deeper into the lipidome using the consecutive runs, each time triggering new precursors for the fragmentation. And for some specific lipid classes, we even had to go for the targeted analysis using parallel reaction monitoring. For the polar and amphiphilic lipids, which we obtained after the second liquid-lipid extraction, we also applied different types of the chromatography, so C18 or C30, and for some of the analytes, it was a helix separation as well. So just to give you an idea how many analyses were done, so it's just using these pooled samples, we performed more than 110 LC-MS runs to obtain this kind of information. Yeah, so this is an example, so you could see how reverse phase chromatography separation looked for the TGs on the C30 or for the polar lipids on C18, and we used helix chromatography here coupled to MS to measure acylcarnitines. So the chromatography dimension was a very important part for us here because we not only used it to increase the peak capacity of our analytical system by providing two separation methods, so LC and mass spectrometry, but we also intensively used that afterwards for the confirmation of our identifications, so annotation of our lipids via retention time mapping approach. So the part with of the workflow when we looked for the lipid annotation was actually very intense, and we spent, I think, more than half a year just on that because we wanted to annotate our lipids on the most accurate way we can with the methods we used, and really that we can rely on every single lipid which we report for the white adipose tissue. And for those of you who are interested in those all annotation rules which we applied, you can go back to our paper published, and in the supplementary file, one contains like many pages describing all these annotation rules and how we perform that. Yeah, so we summarized for each lipid class general fragmentation patterns in positive or negative mode, and in many cases positive and negative mode, as you can see here illustrated for PE lipids. And in this particular case, for instance, we look how we can distinguish acyl, alkyl, and alkenyl linked PEs. So then we have some examples of MSMS spectra for each of the lipid subclass, in this case in positive but also in negative mode. The tables which are summarizing all the fragments which are unique or shared between those lipid subclasses. And finally, this retention time mapping using Kendrick mass defect plots and the retention time to assure accurate annotation of each of the lipid species which we identified. So, and that was done for all lipid classes and subclasses we reported, so that if you are interested in lipid identification, so you're welcome to look at this supplementary file, and I'm pretty sure it will be useful for you. So the next big part which we focused on is lipid quantification, because we were able to report, I think, more than 1,300 lipids molecular species in white adipose tissue. But on this, let's say, qualitative level, it says, of course, something, but not so much to us. Yeah, so we wanted to know not only the physiological levels, but we also wanted to see what happens in the pathology, what happens in the obesity, but also how those 1,300 molecular species are distributed in the white adipose tissue. So that's why the next important point was lipid quantification. And here you can see a general workflow. So to assure this, let's say, high quality of lipid quantification, or the best we can do with the current approaches for this high number of lipid species, we also had to address several challenges on this way. And I would like to share with you this kind of thoughts of what we did, and really would be interesting to know your opinion on that, and maybe exchange of the experience from your side in the discussion part. So the first part was a combination of our internal standard mixtures, yeah, for the quantification. So with the aim of quantifying hundreds of lipids, we obviously cannot go here for real absolute quantification. So we went for the what we call semi-absolute quantification using several lipid standards per lipid class. And knowing the pluses and minuses of this approach, so we tried to take everything of that in consideration. So the first was a design of the internal standard mixture, which will be representative for the white adipose tissue. So of course, there is no standard mixture which is available there just for that, but we tried our best to combine the standards which are possible. So of course, we can rely on already some pre-mixed mixtures. For instance, we used a flash lipidomics mix and also ceramide mix from Avanti. But we also had to spike the additional lipid classes standards based on the diversity of the lipids which we identified. And that, for instance, included different types of acylcarnitines here. So we also went for the fatty acids standards, and specificity of adipose tissue is, of course, high dynamic range of the lipid concentrations, even within triacylglycerols. So if we would use one triacylglycerols present in splash, that would not address this high dynamic range of the concentration of TGs in a white adipose tissue lipid extract. So we had to add some additional one in the different quantities to make sure that we can actually accurately quantify what is going on there. Another class, and I will mention that later on as well, which actually was a surprisingly diverse, is ceramides. And in our identification data, we found much more ceramides than actually we expected to be there, and classes-wise as well. That's why in these mixtures, you could see that we had to add some dihydroceramides, deoxyceramides, phytoceramides, and ceramides with hydroxylation on the acyl chain as well. So that kind of design of internal standard mixtures, this is also the process which requires some optimization. So first of all, we have to confirm which lipids we have in our samples, then address what are missing in already pre-mixed mixtures, and try to add them by using individual lipid standards. The other question is, of course, here the dynamic range, how much we fit, because within our lipid classes, we also have quite high variability. So for that, we actually performed a series of experiments. I'm not showing that here, where we spiked those internal standards in the different concentration in the lipid extracts from white adipose tissue, and try to find the optimal combination of different standards, which would allow us to go further on with samples in this one-point calibration approach, but before proving this linearity ranges and coverage of the dynamic ranges as well. So the next thing is when we obtained our quantitative data, is how we actually integrate those peak areas and what we have to consider. And here also looking into the data in a raw way, not only automated. So we figure out that we have to consider more than one adduct per lipid class. And for instance, if we talk about TG, so what we can expect to be there is maybe protonated, maybe not. Of course, that the ammoniated is the most intense adduct which is present, sediation, and potassium adducts. And the question is how it's important. Does it really necessary to go in all these troubles by quantifying these adducts? So we did that and plotted it in this kind of diagram here, where you can see on the x-axis different TGs listed in the increasing number of carbons, and for each carbon series increasing number of double bonds. And you could see in the different color the percentage of different adducts. So in blue, this is ammoniated adducts, the typical one. And then in red, there is a potassium adduct, and in green, it's a sodium adduct. So even without looking into very fine details which TG is what, you can clearly see that the proportion of adducts changes based mostly on the number of double bonds in your triacyl glycerol. And if we take some extremes, you can see on the right-hand side, so for TG 20 to 24 carbons, 0 double bond, and TG 57 carbons, 6 double bond, the percent of different adducts is really different. Yeah, so in the second case, we have 98% of ammoniated product. In the other case, we have 26% of the sodium and almost 18% of potassium. So if we would not integrate all those adducts together, so our quantification would be not accurate at all. The other phenomena which we have to consider is in-source fragmentation. And for many lipid species, that is quite a big deal. So probably one of the classical examples where this effect is really tremendous is the ceramides. And in ceramides undergo easily in-source fragmentation with the loss of water. And if we would expect a single signal on the MS level from these molecules, that would be a big mistake, because in fact what we see in the positive ion mode, for instance, is a protonated species, is a loss of water, and also sediated one. So in this case, again, we have to perform the quantification in a way when we integrate not only monoisotopic signal from the protonated ceramides, but also the water loss and sodiated form as well. Then this kind of effect is also a lipid subclass specific. And you could see here, again, this kind of plots for the protonated, sodiated, and water loss via in-source fragmentation. And that is plotted for different subclasses. So the oxyceramide, dihydro, typical ceramide, sphingosine, and phyceramide. And again, just by looking on this color representation of adducts abundance, you could see that it really differs between different classes. Yeah? So the oxyceramide, obviously, they don't have a possibility to lose this water. Dihydroceramide already provides significant percent of the water loss. Typical ceramide, this is the one which loses the most, around 21% of water. And for instance, phytoceramides with 3-hydroxy group, for whatever reason, doesn't lose water as well. So that's, again, integration of all these signals was very important for accurate quantification as well. So to make it the story even more complex, even within exactly the same class, but with different chain lengths, you still have a different distribution of adduct and in-source fragments abundances. So the message here is that in order to increase the accuracy of quantification, one really has to combine and these different adducts together. So the other effect, which I guess many people now are aware of, is importance of isotopic corrections to be performed on your data. And in one previous study, we made an example where we looked at what is the input of different isotopic corrections. Yeah, you know about type 1 isotopic correction, type 2 isotopic correction. So we have to correct for the differences in the carbon number between our internal standard and lipid which we are quantifying. That will give a different isotopic distribution. Yeah. We also have to correct for the overlap between lipids with one double bond less. than the one which we are quantifying, and so on. So, and this example shows you that actually the type 2 isotopic correction, depending on the lipid species which you are talking about, can give you a deviation from the real concentration up to 60%. And type 1 correction allows actually to correct for 40 to 55% of the deviation. So putting it up together, these different factors for the quantitative analysis which I mentioned, they account for quite a number of possible inaccuracy in the quantitative data. So with our calculation, we can go with up to 20%, usually lipidome dependent with type 1 correction. 60 can actually go through the type 2 correction. Incomplete isotopic enrichment of internal standards also has to be considered. I don't show the data here, but it's really, if you use the iterated standards especially, it's actually a good idea to look to the isotopologues -1 and -2. In some cases, it can go up to 28%. So ADDUCTS, I guess, I hope I convince you that that is quite an important issue to actually sum up this differential ADDUCTS. And in case of TG, for instance, we could be wrong by 65%, as well as in source fragmentation, for instance, for ceramide, it can account up to 89%, so of deviations. And with that, this is even not covering all aspects of this quantitative analysis, which actually we went through. So another aspect is the response factors. This is another step to come even closer for the accurate quantification. We didn't do it for every single lipid class, as one probably should do, but we did it here for triacylglycerols because that was the most abundant lipid class with the highest dynamic range of concentrations, and we had, I think, only 3 standards for it. So, and for the triacylglycerols, we also calculated the response factor to account for the differences in the carbon number and double bonds into the mass spectrometry response here as well. So with that all performed, finally at some moment we came to our quantitative reference lipidome of human white adipose tissue, which we call ADIPOATLAS. And out of this 1,300 molecular species, we were able to quantify over 700. So we didn't quantify triacylglycerols as a molecular species. So here we made on the species level quantification. And you could see here a presentation of the ADIPOATLAS data, so the concentration of different lipid classes. And the dynamic range, of course, is amazing. It spans over 8 orders of magnitude between different lipids in the human white lipidome, starting with a very abundant TG and going down to very low abundant acylcarnitines. And even within triacylglycerols, the concentration range covers 5 orders of magnitude. And to give you at the end of my talk some examples, actually, what is the use of all the stories, what we found after looking into that, I will just show one example, but there are many of those which we are now digging deeper and deeper into this biology of human white adipose tissue lipid metabolism. So just to show you one example, this is a class of sphingolipids, and here what we were pretty much surprised is to uncover this very huge diversity of ceramides in the human white adipose tissue. In addition to very classical ceramides, which were already measured before in the human white and used as a predictor of the, or not a predictor, or at least associated with the development of insulin resistance, we were able to show that diversity of a species here in terms of the combination of sphingosine bases and acyl chains is much higher than we actually expected to see. And when we looked now for this whole diversity of the ceramide lipids, but we also looked at the difference between lean adipose tissue and obese adipose tissue, we saw a number of species which is increasing with the development of obesity. And in principle, that was not a surprise. As I mentioned, this kind of ceramides with this sphingosine basis, yeah, so say classical ceramides with sphingosine, they are known to be elevated with the development of obesity and often associated with insulin-resistant states. So we could see that as well in our data, that both in subcutaneous and visceral depot, that kind of ceramides went up with in obese individuals in comparison to lean. But what you can also see here, having this absolute quantitative value allows us to see the quite significant difference between subcutaneous depot and visceral adipose tissue depot. And so the level, for instance, of ceramides in subcutaneous obese is just equal to visceral lean, and then it goes even up with a visceral obese tissue. But we also saw a new lipid class, or at least was not reported before in the white adipose tissue, which is sphingosine ceramides. And those ones are formed by the introduction of additional double bond in the sphingosine base. And a few years ago, the enzyme which is responsible for this reaction was assigned, so the SPAT3 enzyme. And when we look on those sphingosine ceramides, it turned out that they are pretty enriched in the human white adipose tissue. You can see that the concentration are pretty similar to the one above. And they provide even higher or even better discriminative power between lean and obese individuals. And this new class, so obviously enrich in the human white adipose tissue and show this most significant increase in obesity, independent on depot. Yeah, so again, subcutaneous is a bit lower, but here we can see a massive upregulation of those lipids with the development of obesity. So another lipid class within the ceramides or subclass was the oxyceramides. And the oxyceramides usually associated with certain lipotoxic effects, mostly due to the fact that they cannot be catabolized through the classical ceramide pathway. And the oxyceramides, so they lack this hydroxy group. And they were reported before to be actually upregulated in the blood plasma and the certain metabolic complications. But what we saw here, that first of all, they're really enriched in the white adipose tissue. So if we compare the published data from the different ceramides based on their basis, sphingosine basis in the human plasma, you can see on this graphs on the left-hand side. So the distribution was quite comparable to the ceramides in the white adipose tissue, except the oxyceramides. Yeah? So they're definitely enriched in the WAT. And what is interesting is that with development of obesity, this level is dropping down. So it looks like WAT serves as a reservoir of the oxyceramides, and upon metabolic complication, they would be released, who knows why, in what, in which way, into the blood plasma, and that would actually feed to the previously published result of the accumulation of the oxyceramide in the blood plasma upon different metabolic complications. So again, here we can see some very interesting metabolic phenomena, which are worth of following up and looking deeper already now from some biochemistry point of view. So again, looking on this whole diversity of ceramides, sphingolipids in the human white adipose tissue makes us think that there is a lot of biochemistry behind which we can discover now, because we know which kind of species we are talking about, and we know also the quantity of those species which are present in lean or obese individuals, also insulin sensitive and insulin resistant states. And this general paradigm about ceramide lipotoxicity in general, in my opinion, has to be actually redefined on the basis of ceramide molecular specificity. So probably different types of ceramides, different molecular species of ceramides might be responsible for either adaptive responses, leading, for instance, to selective insulin resistance, or if that barrier is passed, given to the lipotoxic effect associated with the full insulin resistance. But probably we shouldn't combine all ceramides in one pot, but look at their molecular species with a different view on their functionalities. So with that, I would like to conclude by saying that looking into this human white adipose tissue lipidome was quite an analytical adventure. So we learned many things just by doing. So then probably if somebody wants to characterize different tissue lipidomes, you have to consider at least three points, so that the tissue, so that the lipid extraction and sample preparation in this case really have to be tissue tailored, and in many cases the same methods would not work for the different tissues and organs. So very important point, I think, in the characterizing such reference lipidomes is to invest into the accurate annotation of lipid molecular species. And for instance, the approach which we chose here is really going very deep in the fragmentation pattern and retention time mapping in this case. And I guess I showed you that the semi-quantitative or semi-absolute quantification approach also requires a lot of adaptation and consideration, such as design of the tissue tailored internal standard mixtures, looking for all those differential adducts in source fragmentation products, and also isotopic correction, as well as response factors. So, and with that, I would like to conclude, and I must say that this work was driven mostly by Mike Lange, and he was a person who actually performed all wet lab experiments and also a lot of data analysis and quantification, and discovered all these interesting facts and effects, which I showed you today, but also on the computational side, on the lipid identification and annotation, Georgia and Ni were very, very instrumental. And thank you very much for your attention, and I would be happy to answer your question and actually to hear your opinion about this quantification, identification method. So it would be great to share some expertise here.
Wow, thank you so much, Maria. That was a really, really fascinating talk, and a lot of optimization went into to getting those results. That was really exciting to see. I have one question from sort of the beginning of your presentation. When you talked about the adipose tissue being 98% triglycerides, did you look if there was any difference in TG species between obese individuals with insulin sensitivity versus obese individuals with insulin resistance?
Absolutely. Yeah, I didn't show it here, just like I mentioned, this resource, this ADIPOATLAS, provides so much information that there were some things which were obvious from the beginning and some things which were still digging in. And for instance, address your question, this molecular specificity of TGs, that was one of the things which was pretty obvious for us from the beginning. And I didn't show it here. If somebody is interested, you can read it in the manuscript or in the publication. But indeed, yeah, so in obesity, and especially in the insulin-resistant states, we have upregulation of PUFA-containing TGs in adipose tissue. And since after that study, we measured all that again on the individual samples in this cohort, we also can now correlate it with insulin resistance status, and actually with the hypertrophy of adipose tissue, it seems like this accumulation of PUFAs in those TG leads to increase of the adipocyte size.
Wow, very interesting. It looks like we have a question in the chat from Liang Zhao at Johns Hopkins. As for the lipid analysis with HELIC, what type of column did you use, and how robust were the retention times for different species?
I think we used silica without any modification on that for the HELIC separation, and we actually did it only for acylcarnitines here. We didn't quantify anything else from the HELIC data except acylcarnitines. So for acylcarnitines, it was fine. So it was robust for that one. And just because we could not separate them on the reverse phase, yeah, they are too polar, so they would go into that volume. And otherwise, we mostly relied on the reverse phase because that allows us to separate the isomers, yeah, for the lipids with a different combination of fatty acyl chains.
Yeah, interesting. And just out of curiosity for the carnitines, did you use an available carnitine mix, or did you use individual carnitine standards?
I think we had the individual standards. Yep.
Okay.
It was, I think, in the table which I showed, there was probably 5 to 6 different acylcarnitines deuterated, plus carnitine itself.
One thing that stood out to me was the comparison of the white adipose tissue ceramide levels and the plasma ceramide levels, and sort of the similarity and also the dissimilarity between those two. And I was curious if you think that other organs, how other organs or other tissues might relate to the plasma lipidome as in terms of plasma being a readout for those organs. Sorry, that was a long-winded
I think that is
question.
absolutely a wonderful question, which I would like to know the answer for, because for that, this is probably the point which I tried to make in my introduction then. When we will have the reference lipidomes of different tissues, only then we can actually say what is coming from what, right? Or in some kind of, at least we will have tools to do that. Because there is always a discussion where ceramides are coming from, right? It can be adipose tissue, it can be liver, it can be muscles, yeah? So, and at that level, we do not know at all. But of course, that would be something what would be super interesting to have really know the answer, where all those, markers which we desire to see in the blood plasma, what actually they report, and metabolic status of which tissue and to which extent.
Yeah, great. We have a couple more questions in the chat. Chris Ling wants to know, why did you use a C30 phase for TGs and CEs? Does a C30 phase give better separation for those compounds?
Yes, it does. Yeah, it's not a wow effect, but it's definitely a bit better than C18. And actually now I think in most of our LCMS workflows, we completely switch to C30 columns because just a little bit better separation, but even this a little bit is in many cases making a difference.
Great. Do you experience significant in-source acyl chain loss for CEs? And if so, how do you identify CEs?
Yeah, CE is one of the examples which provides super intensive in-source fragmentation. Yeah, so when you see elution of your cholesterol esters, so you will see there for sure the signal of cholesterol ion. Yeah, so the cholesterol with a loss of water. So it's one of the examples where you have to integrate all the signals. Yeah. And in the LC separation, this is possible because cholesterol itself will separate from cholesterol esters, and cholesterol esters with different acyl chains also will separate from each other. And that's why you can integrate the signals accordingly in this case. But again, here in-source fragmentation is huge. And even so, maybe it's even worse case than ceramides, because we also tried and we published some paper on the in-source fragmentation, optimizing the ion optics, yeah, in front before the mass analyzer, yeah, like tuning different parameters and so on. And it's possible to get pretty down with in-source fragmentation for phospholipids. It also can be done for ceramides to a certain extent, but cholesterol esters is usually the biggest problem indeed.
Yeah, great. Another question, how could you deal with tricky isobaric species where we may have some assumptions? like a PC0341 or PC331, for example?
So in terms of PC 34.1 and 33.1, probably this is a question about like difference in acyl and alkyl chains, yeah. So and then there is in isolipids there is a difference between O and P-link lipids, and again we spent quite a time in looking into that because we were very interested in actually resolving them. And in this supplementary file, which I mentioned from the manuscript, yeah, so if you're really interested in that, I recommend you to look because they do have different fragments, and we have a fragmentation patterns for them in positive and negative modes, but they also have a different retention times, and then a combination of looking at these different fragments and combining that with retention time mapping, that was the way how to resolve this kind of isomers.
Great. Liang Zhao wants to know, what's the most benefit for using C30 reverse phase column as compared to C18? And do you have concern about some lipids that may bind to the C30 column strongly and not being eluted from the column?
Yeah, the benefit is, I think, pretty easy. Yeah, you have a longer chain and stationary phase, so you have a better separation, especially for the species which are like TGs, very unpolar. Yeah, so it's also improving separation for the phospholipids a bit, but the most effects are seen for the neutral lipids in our case. And this is also a good question about if you make sure that everything elutes. Yeah, we try to make sure that everything elutes, and this is something also, I think, a good idea, again, a practical advice which we learned by doing, that if you have a sample which is very rich with triacylglycerols, for instance, or cholesterol esters, you better check that everything is not only eluting, but also eluting in the right moment or time, because sometimes you have a carryover which is not so obvious, yeah, and then by again mapping this with this retention time KMD plots, this is how we actually figure out, for instance, that our gradient, also it was not so obvious, was not fitting first for this kind of application, and then we had to enlarge our gradient for a higher percent B for a while, right, to make sure that all TGs would get out in their corresponding times and so on.
Yeah, really interesting. Michaela says, thank you for the great talk. When creating those reference lipidomes, will there always be a specific extraction protocol associated with those concentrations because of the extraction efficiency from tissues? And will there need to be different extraction solvents that can't be corrected even with internal standards?
Yeah, sure. I think there's many points again to consider here. So we what we actually did, we collected, okay, before we started this extraction optimization, we collected everything what was published before about white adipose tissue, independent human or animal one, and looked at what kind of lipid classes people detected there at all, yeah. Then I will also try some kind of chemoinformatic approaches by calculating the log P's, log D's, and PI's of this lipid to see the optimal solvent system. Then based on this data, we decided, okay, on 3 different liquid-liquid extraction systems, which we're gonna try first. And then we made 3 different extractions. First ones like Folch, MTBE, and then was something else, I forgot. But then from these ones, we use 3 different enrichment strategies. So the liquid-liquid extraction, ethanol, hexane, then the cartridges for phospholipids depletion, or enrichment in this case, and then another kind of SPE protocol, and then all this combination we compared, yeah. So, and we did it not of course by mass spec, because that would be a bit too intense, and we use, for instance, TLC, quantitative TLC in this case, just to see which lipid classes we are covering, to which extent. We also made some NMR for phospholipids. And based on all of this data, we decided, okay, our choice for this tissue, we cover the majority of our lipid classes, would be a fold in combination with ethanol hexane partitioning afterwards. Yeah. But I think if we go for a next tissue, then again, we will try all that again, because considering everything, what like differences of the tissue, starting from the how actually you will disrupt this tissue. Yeah, so we also compared, for instance, these beads which you are using for the tissue lyser, ceramic versus metal beads, and so on, which are because something can get stuck to the beads as well, and such kind of things. And I think with every new tissue, you kind of have to go through that again, but because they will also have different ion concentration and such kind of things. But since we did it already once, first time, of course, it was quite time consuming. So it was like doing and learning from that every time. But now we have protocols which we can just go on, and that would be probably easy. Or easier, not easy.
Chris Ling wants to know, are you able to detect wax esters?
No, we didn't. But maybe, I don't know, they're there, we didn't look for them. Because the problem is with, so wax esters would be also in that fraction with TGs, right? And one of the complications we had after everything what we did is 111 runs, we could not identify any cholesterol esters. And so we said, okay, that could not be, because there are definitely cholesterol esters inside, right? And for detection of cholesterol esters in these very, very TAC-rich mixtures, we went for the PRM afterwards. So for the targeted detection, yeah. Because the signal, so the TG and CE, they are coeluting in this wide range there of species, and TGs are totally suppressing the cholesterol esters. So it was not possible to detect them. We also searched in the same way for retinol esters. No, nothing we found. Wax esters, we didn't look. Maybe they might be there as well, but not visible in such kind of way.
Great. One question that came to mind for me is how homogeneous or heterogeneous is adipose tissue in terms of cell types? And have you tried enriching for different cell types?
Yeah, and that is, of course, another very interesting question. And yeah, we now do the same experiment with tissue which is fractionated or sorted kind of to adipocyte and the vascular fractions, yeah, so where the rest of the cells are. And that's what we want to see, actually, for instance, where this Sphinga D mean ceramides, for instance, do they come from adipocytes, or do they come, for instance, from adipose tissue macrophages, yeah, or something like that. And there will be different patterns, of course, between those tissues. But the problem, the sample preparation for like sorting, yeah, of the adipose tissue, because it's quite complicated, because adipocytes are not so stable when they're heavy loaded with lipids. But again, in collaboration with Matthias Blue and Leipzig, so they gave us some of the sorted samples. So we hope now to map at least adipocyte versus the other cells in the adipose tissue.
Yeah, great. All right, with that, I think we're nearing the end of the hour, and I think we've addressed all of the questions. So I just want to say thank you so much, Maria, for being our first speaker as part of this webinar and giving such a fascinating presentation. Thank you to everyone for attending and for your great questions. And we look forward to seeing all of you at part 2 of Exploring the Lipidome next month. So thank you again,
Thank
Maria.
you very
We
much
really,
for
really
hosting me.
Yeah, absolutely. All right. Take care, everyone.
Yep. Bye-bye.
Bye.
Bye.